All right, let's go through paper 4, which is the extended theory paper for biology. This is from IGCSE, from Cambridge International Examinations, CIE, October-November 2018, and variant 2, so 0610/42 is the code. Let's get started.
Yeah, let's do it. I'm pumped.
Question 1. Wetlands are important ecosystems. Researchers studied the feeding relationships between the organisms in an area of wetland on the coast of Texas. Figure 1.1 shows part of the food web that they studied. Okay, we have some clover, grass, algae, and phytoplankton, muskrat, snow goose, oyster, mysid shrimp, water fleas, pipefish, blennies, spotted sandpipers, stone crabs, marsh rice rats, and bald eagles. And they all live in the same area. All right. A. Complete table 1.1 by giving the name of one organism from the food web in figure 1.1 for each row. Keyword there is one. Only write in one thing because there's more than one, but only write in one. And the first thing we're looking at is a producer. So the producers are the ones that take the energy from the sun and produce the initial amount of food for the entire food web. Okay, and so they are the green things. Okay, so we have the clover, we have grass or algae and phytoplankton. Those are all producers. Everything else eats one of those things or more than one. Okay, so you can write down any of them. I'm going to write down clover. So clover. Secondary consumers. All right, so the order goes, we have producers, then primary consumers, and secondary consumers. So we're looking at this. So muskrats are primary consumers, and anything that eats a muskrat is a secondary consumer. So the marsh rice rat can be a secondary consumer. The bald eagle can be a secondary consumer. Okay, grass, snow geese eat grass. So that's a primary consumer. So again, the bald eagle can eat the snow goose. Algae and phytoplankton, okay. Oysters eat algae and phytoplankton, and stone crabs eat oysters. All right, I go algae and phytoplankton. Water fleas also eat them. So mysid shrimp eat water fleas. Pipefish eat algae as well, and blennies eat pipefish. So any of these options you can write down in the table. I'm going to say, why not a stone crab? So the stone crab. Okay, and now we're looking at an animal that feeds at 2 trophic levels. All right, so muskrats, snow geese, oysters, water fleas, and pipefish, they're all primary consumers and only primary consumers. Okay, marsh rat is a secondary consumer, but bald eagles can either eat the snow goose or the marsh rat. So they can be secondary consumers or tertiary consumers. So bald eagle has 2 colors there. All right. So algae and phytoplankton, we have oysters, stone crabs are only eat oysters, and spotted sandpipers only eat secondary consumers. So spotted sandpipers are not one. Mysid shrimp, they only eat water fleas, and the pipefish is a primary consumer. Blenny can eat a primary consumer or a secondary consumer. So they can either be a secondary consumer eating the pipefish or a tertiary consumer eating the mysid shrimp. So you can write down either a bald eagle or a blenny. So I'm going to write down a bald eagle because they're pretty cool. We saw one the other day. The functioning of ecosystems relies on the cycling of nutrients. Figure 1.2 shows part of the nitrogen cycle. All right, so the nitrogen cycle is a bit more complicated than the carbon cycle or the water cycle, but you still need to know it. Okay, so it starts off, nitrogen gas in the air is converted into nitrate ions, and then the nitrate ions are absorbed into plants, and then the plants use those nitrate ions to change into amino acids, and then the amino acids become proteins in the plants, and then the plants die, so they're proteins in dead plants, and they become ammonium ions, and ammonium can be converted into nitrate. Obviously, other animals can eat the proteins in plants as well, and then it can go into the primary consumers and secondary consumers. But that's not in this cycle. B, state the name of process A in figure 1.2 and give the type of organism that converts ammonium ions to nitrate ions. So ammonium ions goes to nitrate ions in process A. That is called nitrification. Okay, that is the process. This is the process of nitrification, creating available nitrate, basically. And this process is done by nitrifying bacteria. Nitrifying bacteria. Bacteria that do nitrification are nitrifying bacteria. C, describe how nitrate ions enter the root roots of plants, shown by arrow C on figure 1.2. Well, first of all, it's as it says, it goes into the roots of the plants, and it enters the roots through the root hairs. Okay, so it goes into the root hairs using active transport. And the active transport is across the cell membranes, and active transport goes from low concentration to high concentration. So it goes against the concentration gradient. So nitrate ions enter the root hairs by active transport across cell membranes from low concentration to high concentration. Whenever you mention active transport, or passive transport, or even diffusion, you should probably say if it's going from low to high or high to low, or against the concentration gradient or with the concentration gradient. All right. And as it's active transport, it needs energy. So active transport requires energy, or you can say ATP. Okay, some of the nitrate ions will also enter via diffusion. So if there is actually a high concentration outside the root, then they can go in by diffusion. So some is also transported by diffusion. Okay, the first point here has more than one mark. So if you didn't realize that some is also transported by diffusion, you could probably get enough marks from the first couple points. The third point is also true, and you can get a mark for it. D, state the name of the structure in plant cells where process D occurs. So process D, when amino acids in plants become proteins in plants. Okay, that's protein synthesis. Where does that occur? Well, that occurs on the ribosomes. Okay, it occurs on the ribosomes. It says state the name of the structure in plant cells. A lot of the ribosomes are on the rough endoplasmic reticulum as well. So you can call it the rough endoplasmic reticulum as well. That's also another structure in plants where process D occurs. Same thing though, because it's the ribosomes. E, state the process that occurs at B. The process that occurs at B, that is when nitrogen gas in the air, N2, become nitrate ions, NO3-. All right. And that process is called nitrogen fixation. So nitrogen is very, very stable when it's N2, so it requires something like lightning or bacteria to turn it into NO3-, which is available to the plants because the plants can't use N2. F, a pyramid of numbers for the wetland ecosystem show that there are very large numbers of organisms at the base of the pyramid and very few at the top. Okay, so we have lots at the base, then it doesn't say what's in the middle, and few at the top. It could actually be more in the middle. It doesn't say, it just says the bottom and top, but it doesn't matter. It's saying explain why there's a lot at the bottom and not very many at the top. And the reason why is always to do with energy. Okay, there isn't very much energy that can be transferred between the levels. And basically, what effect does that have, and why is there not much energy transferred between the levels? That's what this question wants you to write. So there is very little energy transferred between the trophic levels, so there's not enough energy at the top of the pyramid to support many individuals. It will only support a few of them. So naming some of the ways that energy can be lost. So energy can be lost by respiration, movement, excretion, urine, feces. All those things are wasted energy for the next trophic level. And another way that some energy can be lost between levels, not all the organisms from one level are eaten by the next. So if you have a field of clover with thousands of clover plants, they won't all be eaten by the next level up, or else you would have a completely bare field. Some of the clover will die and be fertilizer basically for the next clover that grows, or whatever other plants grow. Question 2. Figure 2.1 shows a dwarf sunflower and a tall sunflower, Helianthus anus. The height of the dwarf sunflower is 0.45 meters, 0.45 meters, and the height of the tall sunflower is 4.5 meters. Big difference. Dwarf plants, like the one in figure 2.1, have mutant alleles. So the mutant allele is with this one, with a small one. A, define the term allele. Okay, this is one of those definitions you have to know. So an allele is a particular version of a gene. So an allele is a version of a gene. So it can either be dominant or recessive. That's it. Or codominant. B, shoot growth in plants is controlled by auxins. An enzyme in shoot tips converts molecules of an amino acid into auxins, as shown in figure 2.2. So you have an amino acid using a particular enzyme becomes an auxin, and an auxin is a type of plant hormone. And explain, that's a key word there, explain how a mutation in DNA results in an abnormal enzyme which does not catalyze the reaction shown in figure 2.2. So how does a mutation occur in DNA? So basically they're saying that the mutation means that some plants, the dwarf plants, don't get the auxin that makes them grow tall. Okay, so how does this happen? So first of all, you say a mutation in the DNA means basically there's a change in the base of the DNA. So a mutation in the DNA means there's a change in the base in the DNA, a change in a base in the DNA. It can be just one. It can be either added, deleted, or changed. All right. And this base sequence is really important because it codes for the enzyme, which is a protein. So the base sequence codes for the enzyme, which is a protein. So DNA codes for proteins. You can mention something here about the role of mRNA, messenger RNA. I'm not going to right now. It's only 3 marks. I'm going to write down another couple as well. So if you have a different sequence of bases, there's a different sequence of amino acids in the protein. If there's a different sequence of bases, there is a different sequence of amino acids in the protein. And what does this mean? It means that the active site may be different and not complementary to the substrate. This means that the active site may be different and not complementary to the substrate, so the substrate will not bind. If it doesn't bind, then it doesn't create the reaction to create the auxin. All right, so that's 4 marks for a 3-mark question. There should be enough there to get good marks. C, 2 tall sunflower plants were crossed. 25% of the offspring produced were dwarf. Explain how it is possible for 2 tall parents to have this percentage of dwarf offspring. All right, to figure this out, we'll just create the genotype. So we're going to say the tall, because it's dominant, the allele is T, and the short, the dwarf, the allele is little t. Okay, so we have the parents. We have, they are tall with tall. We have the F1 generation is, it can be basically tall plus tall plus tall plus dwarf. All right, tall plus tall plus tall plus dwarf. Okay, this line you don't have to write in normally when you're doing these sorts of diagrams. Okay, so the genotype of the tall has to include 1 gene from a dwarf from each parent. So that means the genotype is capital T little t by capital T little t. And if we do our little square for our F1 generation, we have capital T little t, capital T little t. Sorry, the lines are difficult to draw straight here. That's okay. We have capital T, capital T, capital T little t, capital T little t, and little t little t. And the only ones that will be dwarf will be these, and that means 1 in 4 chance of it being dwarf. All right, so that's how it works. So let's write that in. The allele for dwarfism is recessive and both parents are heterozygous. Remember, you can't write heterozygous dominant because heterozygous means you have one of each. You can't be heterozygous dominant or heterozygous recessive. It's just heterozygous. It means one of each. So what does this mean? This means that the parents do not express the allele but are carriers. Okay. D, figure 2.3 shows how several strawberry plants can be formed from one parent plant. Okay, so what they're cutting, that is a rhizome. That is basically an above ground stem. It's a stem that goes along the ground. And as it's a stem that goes along the ground, if it's cut, then there'll still be new roots that can form under here, and it can form a new stem. And it forms a brand new plant that is exactly the same as the adult plant. This happens in strawberries and many other things. 1. Explain the type of reproduction that produces plants by the method shown in figure 2.3. So explain, so what is it? This is an example of asexual reproduction. There are no pollen or ovules involved in this, so it's asexual reproduction. So the plantlets, that's where you can say a little plant is a plantlet, or you can say the offspring, grow by mitosis, not meiosis. There is no meiosis involved. The plantlets grow by mitosis. And the key bit here is asexual reproduction. So all the new plants are genetically identical. That's the key bit when you talk about asexual reproduction. 2. Explain, again, the disadvantages of the type of reproduction shown in figure 2.3. What are some of the disadvantages of asexual reproduction? The key bit is that all the offspring are genetically identical, so there's very little genetic variation. If there's no genetic variation. There is no genetic variation with asexual reproduction. And what does this mean? There are no adaptive features, so natural selection or evolution cannot occur if the environment changes. For example, if it gets very dry, or if it gets very warm, or if it gets very wet, or very cold. All right. Another thing that happens with no genetic variation is a problem with diseases or pests. So all the individuals are just as susceptible to new diseases or pests. Okay, so there's 3 good marks. Another thing you can say is that there is also a higher risk of extinction because of these, because if you get a new pest, then they can wipe out the entire species, okay, because there's no genetic variation. Also, another thing about this method is that there's more competition for resources because the individuals live closer together. So if the rhizome, the above ground stem, is cut, the new plant grows right there and doesn't get dispersed on the wind or by birds or in water or something like that. It's just right next to the parent plant. So it's competing with the adult plant for resources. Question 3a. Figure 3.1 is a photomicrograph of some xylem vessels. This is quite an interesting perspective of this. So 1, state one structural feature of xylem vessels and explain how this is related to the function of water transport. So one feature that's related to structure, and then you need to explain how it's related to the function of water transport. So don't discuss anything about support. So obviously there's lots of structural features of xylem that have to do with water transport. I'm going to say that the xylem have no cell contents. You don't see a nucleus or ribosomes or any of those things that you'd see or cytoplasm or any of those things you'd see in every other cell, because the mature xylem are dead cells. So you can say there's no cell contents, so the cells are like pipes. Okay. Now, why does this help with water transport? Well, if there's nothing in the way, it means that there's less resistance to the water flow. So this is to provide little resistance to water flow. The only resistance is the water pulling on the sides of the xylem. Okay, other things you could put down, you could say that the cell walls are very strong, very thick, and that's so that they can withstand the tension of the water to prevent them from collapsing inwards due to the hydrostatic pressure. Also, the lignin in the walls is another feature you can use. And again, that prevents a collapse of the xylem and also makes it so that the xylem are waterproof, because you only want the water going in the direction that it should go, not just anywhere. Okay, another thing about the xylem vessels is, as you can see, they are wide vessels compared to some other cells here. They're very wide vessels. How does that help? Well, it means they can transport a larger volume of water. They also don't have any cross walls or any end walls, and that means that, again, that's to provide little resistance to the flow of water. They do also have pits in the sides of the walls, and that will allow water to go laterally. It will go sideways to where it needs to go. All right, so it's waterproof except for in the direction that it needs to go. 2. Explain the mechanism that is responsible for the movement of water in xylem vessels. Explain. Okay, so the process that's responsible for the movement of water in xylem vessels is transpiration. So explain transpiration. Okay, so transpiration really starts when the water in the xylem evaporates into the mesophyll cells. So water evaporates from the cell walls in the xylem into the mesophyll cells. Okay, so this creates water vapor. So this water vapor then diffuses through stomata. So water vapor diffuses through stomata. So because the water has left the leaves, there is a lower pressure in the leaf or at the top, so that the water is pulled upwards basically. So the water pressure is reduced at the top of the plant, so water moves upwards as a continuous column in the xylem. You could also talk about water potential is reduced at the top of the plant. Okay, so it's a continuous column being pulled up the plant because there's less pressure at the top. And how is it pulled up? Well, because there's cohesion between the water molecules. So the continuous column is due to cohesion between the molecules, and cohesion is basically forces of attraction between the water molecules. Okay, and this creates a transpiration pull. That's what it's called. This is a transpiration pull. The water is being pulled upwards. And the water enters at the bottom and leaves at the top by osmosis down the water potential gradient. So water enters in the root by osmosis down the water potential gradient. Okay, that's a lot more than 4 marks, but that's all good information you could put in. Okay. 3, state one role of xylem vessels other than transport. So xylem don't just move the water, they also support the plant. B, the rate of transpiration is affected by several factors, including the temperature and the humidity of the air. State and explain, which means you need to link them, the effect of an increase in temperature on the rate of transpiration. Okay, so when you increase the temperature, what happens to the rate of transpiration? So when temperature increases, the rate of transpiration increases and then decreases. So as it gets warmer, transpiration will increase. Then you'll get to a temperature that's too hot, say in a very hot desert, and the plant does not want to lose more water, so it actually closes the stomata so that the transpiration decreases. If you just said the rate of transpiration increases, you should get the mark there as well. Okay, and how does that happen? So as transpiration increases, the stomatal pores become wider and the guard cells become turgid. So remember, when the guard cells become turgid, that means it's like a balloon with a thicker side on the middle. It will curve outwards. You have 2 balloons, 2 long balloons with a thicker side in the middle. They'll curve outwards, and that happens when they're turgid, when they're full of water, and so that means the pores open. Okay, so as the transpiration increases, that means that there is more water vapor evaporated from the xylem, and so there's more diffusion of the water vapor out of the stomata. So more water vapor evaporates, and there's more diffusion of water vapor from the stomatal pores. So again, as temperature increases, water molecules have more energy. So if they have more energy, they move faster and you can have that. That's why you get more evaporation. So as temperature increases, water molecules have more energy. So you've stated the effects of increasing temperature. So when it increases, the rate of transpiration increases and then decreases. As temperature increases, there's more energy. That can also be explaining. And you've explained because the stromatopores have become wider and more water can evaporate and diffuse. So you've stated and you've explained. Question 4. Insulin is a hormone that regulates the concentration of glucose in the blood. A. Define the term hormone. Okay, another definition you need to know. So hormone is a chemical produced by an endocrine gland, carried by the blood, and alters the activity of a specific target organ. So a hormone is a chemical substance produced by an endocrine gland, carried by the blood, and alters the activity of a specific target organ, or sometimes more than one. So marks here, it's a chemical produced by an endocrine gland. 1, it's carried by the blood. 2, alters the activity of a specific target organ. 3. In old exams, you might see an endocrine gland written basically as a ductless gland. Okay? And a ductless gland means basically the blood just flows right through it. It's not like the salivary gland where there's a duct that the saliva flows into the mouth with. Okay? An endocrine gland is a better answer because endocrine basically means it produces hormones. B. 2 people, A and B, visited a doctor to discuss their similar symptoms. The doctor thought that their blood glucose concentrations were not very well controlled. A glucose tolerance test was carried out on both people. A and B did not eat or drink anything other than water for 8 hours before the test. They then drank a glucose solution. Blood samples were taken at 30-minute intervals. The samples were tested for glucose concentration. Okay, and the results are shown in figure 4.1. All right, so this is figure 4.1. You can't see the label for the y-axis. It says blood glucose concentration in milligrams per 100 centimeters cubed. But so they, just before the test, they had their blood glucose concentration taken, and it was about the same as it was when the sugar solution was taken, and then different things happened. All right. 1. Use figure 4.1 to state the blood glucose concentrations of A and B at 180 minutes. All right. Okay, so at 180 minutes, we have 2 values. So if you look at this, you go up by 5 squares, and that means you go up by 20 milligrams per 100 centimeters cubed. So 20 divided by 5, so that equals 4 milligrams per 100 centimeters cubed per square. Okay, so every time you go up 1 square, it goes up by 4. So if we look at A, it's halfway between these 2 squares. It goes here. So that is 200, 204, 8. Halfway through, it's 210 milligrams per 100 centimeters cubed. And B, okay, is, go across, use your ruler, make sure you do it carefully. If you look at that, that's 80, 84, 88. That's 88 milligrams per 100 centimeters cubed. So that is 210, and that was 88. It already has the units in there. 2. Calculate the percentage increase in the blood glucose concentration in person A, only person A, between 60 and 90 minutes. All right, give your answer to the nearest whole number. Okay, don't forget that whenever it says that. Show you're working. All right, so person A between 60 and 90 minutes. So at 60 minutes, person A, which is the X, had right here, that is 60, 6, 2, 4, 6, 8, 10, 12, that's 72 milligrams per 100 centimeter cubed. And at 90 minutes, person A had, all the way up here, they had, go across here, it's halfway between the 2 squares. So that is 170. 2, 4, 6, 8, 9, 10. So 170 milligrams per 100 centimeters cubed. So it went from 72 to 170. Okay, so the percent increase equals, It says calculate the percentage increase. So we want to know how much it increased. So the increase, how much it increased, divided by the start times 100 to make it into percent. So the increase was 170 milligrams per 100 centimeters cubed minus 72, which is what it started with, divided by 72 times 100. So if you put that into your calculator, that will equal 136%. So it's 136%. So it went up a lot. 3. Describe how the response of person A differs from the response of person B in figure 4.1. Okay, you're just describing it. And with this, they're saying in figure 4.1, they probably want you to use a data quote. But there's only 2 marks, so you don't have to necessarily include it. It's a good thing though as well. But there's lots of information you can put down. So if we look at this, person A, the X's, increased much more quickly than the increase in person B. Okay, it's a steeper curve. And you can also mention that when the decrease occurred, so when it decreased going back closer to normal, person A also decreased in a much more steep curve. Okay, much more steeply. You can also mention how the maximum blood glucose concentration for person A was definitely higher than the maximum blood glucose concentration for person B. Okay, and person A reached their maximum blood glucose concentration much later. And person A did not return to the starting concentration in the 240 minutes that the test was taken. Okay, as I said, you could do a data quote, but that's 4 good marks there. Okay. So the response of person A increased faster than person B. And you can also mention about how it decreased at a much steeper rate. So the maximum blood glucose concentration of person A was higher, and person A reaches a peak much later than person B. You can also say that person A doesn't return to the starting concentration, and you can mention a data quote with the units. If ever you do a data quote, you have to put in the units or else it doesn't count. Okay, there's 3 marks for a 2-mark question, you're fine. Now is the explain bit. Explain the results of the glucose tolerance test shown by person B. So person B, that's the one where it basically did what it should do. So when the blood glucose concentration increases, it is detected by the pancreas, which releases insulin into the blood. And what does the insulin do? It stimulates the liver to take in sugar and convert it into glycogen using enzymes, and it stimulates muscles to increase the respiration, to take in sugar and increase the respiration. So insulin stimulates the liver to take in glucose and convert it into glycogen using enzymes. Remember, glycogen is a storage form of sugar, glucose, in humans. Okay, it also stimulates the muscles to take in sugar and increase respiration. So the blood glucose concentration decreases back to a normal level. And you can get another mark by mentioning something about homeostasis or negative feedback. Well, describing it in context and properly. All right, good. 5. The doctor thought that person A had type 1 diabetes. Describe 3 symptoms of type 1 diabetes. Okay, there's lots and lots and lots of symptoms of type 1 diabetes. Some are more severe than others. So first of all, it says only 3. Only put down 3. So someone with type 1 diabetes is thirsty all the time. So they drink lots and lots of water. Okay? And they're likely to have weight loss. Okay? Even though they might want to eat more. Okay? And their breath is often very sweet smelling. I've heard it described as musty apples. Okay? You can write those in sentences as well if you like. Okay? And other symptoms you can put down, they can be very tired or very fatigued. They might be very breathless because they're not getting the cue to do as much to store the energy as glycogen or to increase the respiration. They might be very dizzy. They might faint. And if it's severe enough, they might go into a coma, which is very severe. Okay, they will have frequent urination. And if you're willing to try it, it's sticky or sweet urine because sugar goes into the urine and it's not supposed to go into the urine. And because there's sugar in the urine, they're more susceptible to things like urinary tract infections and have recurrent thrush. And thrush is a fungal infection. Okay, because they're thirsty all the time, they might have a very dry mouth. They're also very hungry because their body isn't getting the cues that it needs, that it can store the energy. Okay, and they might have some changes in behavior. They might become very irritable because they're very tired and hungry. And all the other things, they might have mood swings, they might be confused. They're not getting enough energy in their cells to be able to think clearly. There might be nausea or vomiting. They might have blurred vision or blindness if it's severe. And cuts, grazes, wounds, they don't heal very easily. So there's lots and lots and lots and lots of things you can put down, but make sure you only put down the 3 that you're most comfortable with, that you know are accurate. All right, that's the end of this question. Let's move to the next. Question 5. Figure 5.1 shows a photomicrograph of human blood. Okay, so it doesn't quite show perfectly. This here is a phagocyte. Just doesn't show up on the screen properly. This one is a lymphocyte, and this is a red blood cell. And if you look at these little specks here, those are platelets. All right. A. Describe the differences in appearance and the roles of the 3 cells labeled in figure 5.1. Okay, so we want to see differences in appearances, not just describe what one they look like, but compare them to each other. And we want to see the roles. Okay, we don't need to know about the platelets because that was not one of the ones that was labeled. All right, let's start by looking at the red blood cells. Okay, so all of these are red blood cells. This is a red blood cell. This is a red blood cell. And we have some other ones as well. Now, if you notice, they look like they have a light bit in the middle and a dark bit around the outside. The light bit in the middle, dark bit around the outside. That's because their shape, it's a biconcave disc, and it's thicker on the outside than on the inside. So all these little bits, these are hemoglobin that stop the light. There's more on the outside than on the inside, so it looks lighter when the light shines from above. You can see it looks lighter in the middle than it does on the outside. Another thing you'll notice about the red blood cells is they're certainly smaller than the lymphocytes and the phagocytes. Okay, so the phagocyte is much larger than the red blood cell. All right. So let's write this down. We have our red blood cells. And they are smaller than the white blood cells, the lymphocytes and phagocytes. All right, and another thing about their appearance, because I'm describing the appearance first and then I'll do the role. The appearance, they are biconcave discs with no nucleus, and the other 2 clearly have a nucleus. So they're smaller than the phagocytes and lymphocytes, and they're biconcave discs with no nucleus. Now their role. The role of the white blood cells, the red blood cells, is to carry the hemoglobin so that they can transport oxygen through the body. So it contains hemoglobin to transport oxygen. All right, now we're going to talk about the lymphocytes. So obviously they're larger than the red blood cells, we've already mentioned that, but they also have a very large nucleus. Now if you look at this, this is the outside of the cell approximately, and inside here is the nucleus. So the lymphocyte has a very large nucleus with not very much cytoplasm. Okay, and what does it do? Well, the lymphocyte is responsible for active immunity because it creates antibodies in response to antigens. So the lymphocytes have a large nucleus with a little cytoplasm. What do? They're responsible for active immunity by responding to antigens to produce antibodies. All right, now the phagocytes. So the phagocyte is also large, just like the lymphocyte. Sorry, that's much larger, but it has a lobed nucleus. It's very different to the nucleus of pretty much anything else. Most nuclei are circular. This one's lobed. So the phagocyte is about the same size as a lymphocyte with a lobed nucleus. And what does a phagocyte do? Well, it does phagocytosis. So the role is that it engulfs pathogens in the process of phagocytosis. Remember, it does not eat pathogens. It doesn't have teeth. It doesn't have a digestive system. It's just one cell. Okay, it engulfs the pathogens. That's one of the things that the examiners don't like to see when you write the phagocyte eats pathogens. They just don't like that because it's not true. B, figure 5.2 shows some of the stages of blood clotting. So a blood vessel breaks and then platelets collect at the break in the blood vessel. And after that, platelets release an enzyme, and the enzyme converts prothrombin into thrombin. And the thrombin does something, which does something else. And that something else forms a mesh at the break in the blood vessels. I think you know what you're going to do next. 1. Complete figure 5.2 by filling in the 2 empty boxes. Not a surprise. Okay, so we have prothrombin is converted into thrombin, and thrombin triggers the change of fibrinogen, which is a protein, into fibrin, which is basically a mesh. They're fibers. Okay, it's a protein in a mesh. 2. State 2 roles of blood clotting. Okay, blood clotting does some very important things. It says state 2 roles, only state 2 roles. I'm going to put down 3. Okay, one of them is the biggest one really. Blood clotting prevents the loss of blood. Blood clotting prevents blood loss. So if you get a cut, you need blood clotting so you don't lose all of your blood. And another thing it does is it stops things from being able to enter your bloodstream. So to prevent entry of pathogens and microbes. Okay, well, microbes being pathogens really. And another thing it does is it helps in the process of healing the wound. To help in wound healing and tissue repair. Now, when it says state 2 roles of blood clotting, what would happen if you wrote down 3? Well, 2 possible things could happen, and it depends on the particular question. Sometimes they will mark the first 2 regardless of what the third one you've written down is. That's often what they do. Other times, they will mark the first 2, but they'll also glance at the third one, and if the third one is wrong, it cancels out one of the marks. So you never really want to write down a third one unless you are sure, very sure that it's right, because they're not even going to look at it, and if they do, they might take a mark off if it's wrong. So only write down 2. C. Hemophilia is a sex-linked blood disorder in which blood takes a long time to clot. Figure 5.3 is a pedigree diagram to show the inheritance of hemophilia. So what you can see from the key down here is that the males are square and the females are circle. And there's no females with hemophilia, and the males with hemophilia have the black square. Okay? And you can just trace what is happening with that information. The normal allele is represented by X with a capital H, and the mutant allele is represented by X with a small h. So that means it says that this is a sex-linked characteristic, so it's on the X chromosome. It's not on the Y chromosome. 1. State the genotypes of the people identified as P, Q, and R in figure 5.3. Okay, so they want this person, and this person. Okay, so as we said, squares are male and circles are females. Okay, so let's look at person P. Now, person P is female. Okay, she has normal clotting time. All right, so that means that she has to be, as she's female, she has to have 2 X's. That's just how you become female, as you have 2 X's. All right, so now you have to if they have normal clotting time, they have to have at least 1 dominant allele. Okay, but what about the second one? Is that going to be dominant or recessive? Now, if you look at their 1, 2, 3, 4, 5, 6 kids that they had quite a few kids. They had one with hemophilia, and one of their grandchildren had hemophilia. That means that these two had to have an allele for hemophilia from the mother. And I'll show you why in a minute. So it had to be from the mother. So that means that this has to be a small h. She has to be heterozygous. So we have big H, X with a big H, and X with a little h. She's homozygous. Not homozygous, she's heterozygous, sorry. All right, so now we're looking at this person Q. All right, now, first of all, male. He's a male, so he has to have X, Y. Now, this is a sex-linked characteristic, which means that it's only the allele for this characteristic is only on one of the chromosomes, either X or Y, and we've been told it's on the X. So that means this person has to have a recessive gene on the X. Has to be X with a little h and Y, because the Y has nothing to do with whether they have hemophilia or not, which is why the Y came from the father, and the X, that had to come from the mother. So that's why she must have a little h, okay? Because the father cannot give an X, or else the offspring is female. All right, because the mother cannot give a Y. All right, now so this person is X small h Y. Okay, so X small h Y. All right, male with hemophilia. And now person R, that's who we're looking at now, is also male, so it has to be X Y, but they do not have hemophilia, which means this has to be capital H. Okay, that's all that it means. Y again has nothing to do with it. So X, capital H, Y. That's a capital. There we go. All right, and that's all it needs to know. 2. The couple S and T are expecting another child. What is the probability that the child will have hemophilia? All right, so let's look at couple S and T. All right, so couple S and T. So the female, so this is X, X, and this is the male, X, Y. Okay, now for these children, the father has given the Y to this offspring, so that means that's fine. But this boy has hemophilia, which means again there has to be, she doesn't have hemophilia and he doesn't have hemophilia, but she needs to have the recessive allele, so he must be, this must be X, Y with a little h, as we worked out up here. Okay, so the father has a dominant allele and a Y, and the mother has a dominant and recessive allele. No Y's. All right, so if we look at the mother, if we're going to do our little Punnett square, we can put our mother on top. X, capital H, X, little h, because she doesn't have hemophilia. And the father can go at the side. X, capital H, Y. He doesn't have hemophilia. All right, so I put in some nice boxes, nice cells to make this neater. All right, what do we have? The offspring will have X, X, capital H, X, capital H, X, little h, X, capital H, Y, and X, little h, Y. All right, so this is the result, and it says, what is the probability that the child would have hemophilia? This is the only child that could have hemophilia. The probability is a 1 in 4 chance. So you could say 1 in 4, or you could say 25%. All right, so those would be fine. All right, another way you could write this, you can say X, H, XH by XH Y. All right, so this is just another way of doing the same thing. So we can have XH by X capital H. So this is XH, XH. Or we could have XH by Y. So that's X capital H, Y. Or we can have X little h by X capital H. So that is X capital H, X little h. Or finally, the other option is we can have X little h by Y. X little h, Y. So those are the four options. Okay, and again, same thing, 25%. It's just two methods of doing the same thing. Some people learn one way, some people learn the other way. They're both absolutely 100% fine to do, whichever one you're more comfortable. 3, define the term sex-linked characteristic. Okay, a sex-linked characteristic is a gene for the characteristic that is located on a sex chromosome, either the X or the Y. Generally, we talk about the X ones, but sometimes there are Y ones. The gene for the characteristic is located on a sex chromosome, either X or Y. Generally, we talk about X, but it doesn't have to be. And the characteristic is more common in one sex than in the other sex. And most of the time, it's more common in males than in females. 6. Figure 6.1 shows the Galapagos iguana, Amblyrhynchus cristatus. All right. A1. State two features that are used to classify animals, such as the Galapagos iguana, as reptiles. Not necessarily features visible in the diagram, they're just there to make the exam more interesting, I think. But you need to know, how do you classify iguanas as reptiles? Well, 1, reptiles have dry, scaly skin. Dry, scaly skin, as compared to amphibians that have moist skin. Okay? They also have soft-shelled eggs, or leathery eggs. So soft-shelled eggs. You can't just say they lay eggs, because so do birds. So do fish. There's lots of things that lay eggs that are not iguanas, or reptiles. 2. State two features that are present in plant cells that are not present in the cells of reptiles. So reptiles are obviously animal cells. Plants have a cellulose cell wall. So plants have a cellulose cell wall. Animals don't have any cell wall at all, but they certainly don't have cellulose. And plants have chloroplasts with chlorophyll. Chloroplasts with chlorophyll. You could just say chloroplasts. You could just say chlorophyll as well. Those are fine. Another couple things that plants have that animals do not have. Plants have starch grains, or granules, and they have a large permanent vacuole. Okay, remember, animal cells, they have temporary vacuoles for certain purposes, and they are a lot smaller. But plants have the large permanent vacuole that helps with keeping the plant stand upright. Okay, B. Galapagos iguanas feed on seaweed which contains starch and other carbohydrates. State the name of the enzyme that digests starch, and that is amylase. Amylase. 2. State the names of two parts of the alimentary canal where starch is digested. Well, one of them is in the mouth by saliva, and the other is in, because the amylase is in the saliva, and the other is in the small intestine. And that's why the bile needs to be added to the small intestine, because the pH of the mouth is about pH 8, and the pH of your small intestine has to be pH 8 as well, and the stomach acid is very strong acid. Okay. C. There are many threats to wildlife in the Galapagos. Describe ways in which endangered species can be conserved. Okay, this is one where there's a lot of possible answers, There's 5 marks, so you need to put 5 full answers down, but there's a lot of things you can write down. Okay, one of them is that they can monitor the population size by tagging individuals. So monitor population size by tagging individuals. Now, if they monitor the population size, they can tell whether or not it's becoming really critical or whether they're getting more numbers in their population and becoming less of a concern. Okay, another thing that they can do is protect and restore the habitat. So they can protect and restore the habitat. They do that in the Galapagos by not letting many people go to the Galapagos. Okay, another thing they can do is to reduce and prevent pollution, any type of pollution. That, say, if they're water animals, then they can try and reduce the amount of water pollution or prevent certain water pollutions from taking place. Another thing that can be done to help conserve endangered species is to put in place things like hunting bans or specific times that they're allowed to be hunted. So they can have a hunting ban. And another thing they can do is they can create exclusion zones or reserves so that they're not disturbed by people. Okay, that's 5 marks. I'll just go over some other possible answers you could put down. You could say that they can work to remove alien species or prevent colonization by alien species. There can be government legislation put in place in order to protect the species. And along with government legislation, you can have international agreements so that they don't have trade in some of these endangered species. You can specify specific times or areas where hunting is not allowed. So if you can't control hunting and do a complete hunting ban, maybe they could have certain areas or times of the year where hunting is not allowed. All right, they can also remove some of the animals to zoos. And in zoos, they can do quite a few things to help them. They can do some captive breeding programs where they encourage the animals that have different genetics to breed, and then they can possibly release them into the wild, so reintroduction programs. They can also collect either if they're endangered plants, they can put them into seed banks, or if they're endangered animals, they can put their sperm or egg into frozen zoos. And another thing that can be done in order to help conserve endangered species is educating the public, awareness education basically, into how they can either help protect the species or why they need to not poach or pollute certain areas. So again, there's lots and lots of different answers you can put down. Only put down 5 of them though. And D, one aim of conservation is to maintain resources in natural ecosystems. State 3 resources that natural ecosystems provide for humans. Okay, there's lots of things you could put down. One thing that natural ecosystems provide is food. Another thing that natural ecosystems provide are building materials such as timber. So building materials, timber, rocks, that sort of thing. Another thing, water. All right, we use natural water, okay, from lakes and that sort of thing. Underground wet reservoirs. Okay, natural ecosystems can also provide drugs or medicines. Oxygen, that's another thing you could put down because trees produce oxygen. You could name a mineral that's produced naturally in the ecosystems. Ecosystems can provide clothing. In many different ways you can have clothing from natural ecosystems. And this is one where you can have any other valid point. There's lots of little things you could put down. All right? Oh, but only put down 3 again. And that is the end of this exam. I do hope you found that useful, and I hope you'll do really well on your final exams. And if you have liked the video, please press the thumbs up button, and we'd really appreciate it if you'd subscribe if you haven't already. If you have any comments or anything else to say to us, please write them in the discussion section below. We'd love to hear from you. And have a great day.